We're seeing bottom in sight.
All right, there's bottom.
And we are seeing our first look of the beginning of dive number 16.
So I'm just going to hang tight here.
This
Let you
is
get down a little more.
Windows to the Deep Expedition 2018.
Okay, I'm just taking
Our
this
second to last dive.
Sonar in one.
Copy that.
We are at a water depth of almost 510 meters.
Better.
Pilot, you happy with 50 meters on your sonar?
Yep. So what am I at?
And we are in a
So it looks like you're
unnamed canyon that we are calling Pea Island because that's the closest point of land. And that's Pea Island in the Outer Banks of North Carolina.
A little for you here. But you're still 10 meters, 8 meters off bottom.
yeah, it started coming up. I'm coming back down. I was adjusting my
And a
Z
very
bias.
active water column, if you've been watching the last few minutes. Lots of food and lots of predators. We're seeing the snipe eels. We actually inadvertently caught a snipe eel yesterday. I think it was caught up in a piece of Velcro.
On the top of the D2 ROV. And snipe eels eat these surgested shrimps that we see all over the water column. Those shrimp with the red backs.
And we actually could see a shrimp within the body of the snipe eel.
Mm-hmm.
Little pink band. So if we see any with
So you don't want to go to zero.
pink
then you're gonna
spots
start
midway,
rising
we'll
at
know what
15
that
meters
is.
a minute,
Yep.
Or 10 meters a minute. So make sure you
We
So
have
it's
little
keep that tuned in.
barracuda,
positive
which
20
are
or
the
so?
silverfish that you see
Positive
right now.
26 or 7 or 8.
26.
Yeah,
Very
something like
quick
that.
swimming with
I
their
think is where they've
caudal
been
fin.
setting it, although I haven't set it in a while, so I'm not sure.
And here's a great view of a snipe
Yeah,
eel.
lately.
With a pink belly, maybe. It's hard to tell.
Looks,
if that's
yeah, kind of
pink.
looks like
yeah.
it. Yeah.
It's like you
Okay, that
might have had a
seems little
couple.
Heavy.
Yeah, look how bulbous. He's bumpy. Wow.
Yeah, he definitely
Okay.
had
So it's
had
just
quite a
pretty
meal
good.
Tummy
recently.
full.
Fire bottom, even if you're by
He's got to
yourself,
swim it
you'll
off.
get
He's
the
working
information.
out now.
It'll
Okay.
make your life easier.
It was very interesting
Okay.
to put the head of the snipe eel under
down
a bit of
to
a
delta
microscope and
at
look
least
at its
15.
teeth, which
I'm right on the- I'm
basically
just a couple
slant
meters above the
backwards
bottom right
towards
now.
its gut. So
I'll give you
once they
little
get a
more there.
shrimp in there, it has a really hard time
Altitude
escaping.
is
Bridge, this is ROV now.
almost 25 now.
I would like to inform you that the vehicles are on the bottom. I'd also like to verify that center rotation is set around the A-frame, and
I
when you
will
get
switch that now.
So basically,
And when you get a chance, we
what
could
we're seeing in
discuss
the
the
water
bell
column
outlet.
here
Okay, center rotation
is
has been
the
shifted. ready for
midwater
bailout.
fauna.
excellent, the
And
bell
they're
out will be
actually
to the
quite close
northeast
to
if
the
possible.
seabed
Okay.
here.
Bailout
I'm locking
set
in
to
my auto heading.
northeast, and
And
That sounds
the
great to me.
chain
Excellent, sounds good. Thank you very much. Chris.
heading is
of
paged as well.
jelly-like organisms on the left is a salp. No, I mean, it's a siphonophore. Again, another midwater animal.
Looks like streaming. Looks like I'm going off to your.
And
that's
these
okay. Just
animals
keep
are
Okay.
vertical
All right, I'm backing
migrators,
down.
so they'll come
I'll come around
down
and get you.
towards the seafloor during the day when the upper water column is lit so that they can avoid predation, and then they move up in the water column to feed at night when their predators might not be able to see them as well.
Still backing.
And that's called the deep scattering layer. And it provides an ample opportunity for the benthic organisms to have extra
Still
food
backing.
delivered when this layer is close. Oh, and
We are settling right into an area.
so one of the main reasons that we're diving here today is because there have
Okay,
been,
I'm still backing.
it's been detected
Copy.
that there are bubble plumes in the water column, which might
Wind up
be
face down.
indicative of, well, which
Gotcha,
is
view.
indicative of
Copy.
seepage from the seafloor, seepage of methane.
Still backing.
And what we see on the seafloor when that happens are bacterial mats, are one of the first things that
Video, are
we
you talking
see.
to me?
And these white and gray spots that you're seeing on the seafloor is are bacteria that basically
Sending a
use
little
that
bit,
methane.
Stay on the bottom.
And in a couple previous dives, yesterday's dive, we found some unexpected patches like that weren't on any maps. And several dives ago, we actually found some that had bubbles coming out of them. So we're hoping to see more of that today.
5 or 6
somewhere. We have
coming down.
Several sites that had been
Got you right at
mapped
42
using
degrees
some sonar
setup.
evidence that they might be down there. So we're hoping to find those and hoping that we'll find some that are
Okay,
active.
I'm not backing anymore. I'm descending.
It's hard to tell the difference between a self and a line out here, but those are all selfs. Self chains. Coming down slow
Is someone
with you.
trying to call in? We
Copy.
can't quite hear. It sounds kind of muffled.
Are we going to do... Oh, we did white balance already.
There's a snipe eel that had
Another
another
one.
wheel.
We did. I noticed the temperatures dropped.
Plenty to
3
eat.
I wonder if
degrees
these are
when we did it.
Some kind of
Almost 5 degrees since we did
tube
it.
worms that are making these straw-like things that we're
Yeah,
seeing.
yeah. Definitely seeing some eel pouts
Look at
Okay.
this weird fish in Syria.
sitting on the
Never
bottom.
seen
We've
that.
noticed a lot of them in the last several dives, and yesterday and the day before we were noticing that they
Okay, I'm
were
on bottom.
concentrated around these bacterial maps.
Positions look good.
Okay.
I think
So
we're ready
we're
to
wondering
start
about
the pilot.
their association.
Carolyn, is that you?
Yeah, I'm sorry. I'm having technical problems here with the wireless I was using to communicate. Amanda, I don't think she's on the line, the audio, but she also noted that Sentry dove here last year on the cruise that she was on. So there has been a bit of ground truthing here, and so an expectation that we should find something.
Yeah, that's great. Thanks for adding that.
It's like some of the fish are going nuts and
There
stirring
have been,
up,
I guess this
some of the
data
sediment.
originated from Okeanos Explorer and Carolyn Ruppel, and has been coming back to this area and working more extensively. I don't know if you want to talk about that a little bit, Carolyn.
Well, I think we're waiting to see something in the seafloor here. I mean, the point I was trying to make, though, is that Amanda, of course, Amanda Demopoulos at the USGS and others were on a cruise last year,
so what's our initial
and
move
they
gonna be
dove here with
now,
Sentry.
just for
So there has
planning?
been some visualization of the seafloor close to this
Yes.
area
Sorry,
before. What I can
repeat
say is that
what's
the
our
original
initial
plumes
move
were
gonna be?
So
found
the initial
by
move will be heading
Okeanos
towards
Explorer
waypoint 2,
along
which
this
is
margin,
to
and
the
I pulled
southeast
the track
of
lines
us
this morning, but I don't remember the years. I'm sorry.
Um,
Between about 2011 and 2013,
Right.
Yep,
there was
that
a Nature
agrees.
Geoscience publication that was published that included a database. Adam Sparky, who was
It'll
at
be
NOAA
at
at the time, was the
a,
lead author on that.
bearing of a
So in that paper, we
133.
published a database that identified
that was
these
needed.
seeps amongst
If you wanna come around
550
145,
or more other
or,
seeps along the
come
margin
forward, and I'll
for the
line
first
up 145,
time.
and then we
What
can
we've
start
been doing at the USGS
that way.
Copy
since
that.
then, at least in my group, the
So you're
Gas
gonna
Hydrates
start out
Project
at 20
is
meters,
exploring a lot of parts
to
of
begin,
this
or
margin,
do
and
you
in
guys
doing
feel
so
uncomfortable
have
30?
used
I came
instrumentation to
off,
find either new plumes or to verify ones that we already knew existed in
I
that
have a feeling there's
2014
gonna be a lot to
database.
look at, so
And of
maybe
course,
20
people like
and
Amanda
then go from
Demopoulos,
there.
Eric Cortez, and others
Yeah,
have
we
been
definitely
very
wanna do
active in
at
coming
least 10,
back
just
to,
to get us like
and
a little bit
Cindy
closer to
Vandover
what's
has been
going
very
on.
active in
Copy
coming
that.
back
I
to
will
these
move us
places
20
with
meters.
Alvin or Sentry
Just
or
stand
other
by on that for a
vehicles.
second. Let's
We
get
also
lined
had
up.
an ROV cruise last year that was run by the USGS for the north of the margin
Starting to walk
to
my
really
head in
try to
45.
ground truth
Okay.
what's at the base of these water column methane plumes, and especially for the, Cheryl, you know better than
I
I,
don't
for the
think it's
domestic
me. I think it's
ecologists,
the
this
fish.
is
But
of
as
great
soon as I
interest,
get
obviously,
close
to think
to the
about
bottom,
how
they
these
just start
communities
going
have been
nuts and stirring
colonized and
everything up.
developed, and
Yeah.
how they're what the distribution of
compare, I saw
organisms at
them
the seafloor is
jetting down and
relative
swishing on
to
the
the
bottom.
underlying plumbing system.
So, anything extreme
Yes,
with the
that's very interesting, Carolyn, and I hope that we will see some organisms. The last two dives where we have found evidence of seepage, we haven't found extensive
Big jelly?
chemosynthetic communities, and that's one of the things we're looking for when we come to these seeps
Hold
and
on,
finally
still
get
kinda moving pretty quick.
visualization
All
of
right, you can go in.
the seafloor in these areas. see whether there is active seeping.
Okay,
We would
come back
detect
out.
bubbles. We would be able to see bubbles emanating from the seafloor, or if the
Looks like you're gonna be photobombed all
bubbles
all
have been
morning.
emitted
Yeah.
for an extended period of time, we might find chemosynthetic communities that can exist there that rely on the methane that's seeping from the seafloor.
Okay, I'm kind of
And
lined up serious
that
now.
basically translates to food for them, because microbes can use that methane, and then other organisms have microbes within their bodies, symbionts in the gills, that can also use that methane and convert it to food. And so these communities are very unique because they are not reliant on surface productivity that's driven by photosynthesis, like all of the algae, the microscopic planktonic algae, which drives a lot of the ecosystems of the deep sea. These chemosynthetic communities do not rely on the sun's energy, but rely on the energy produced by the methane leaking from the seafloor.
Okay, I'm at 165. That looks kind of uphill to me. You seem lined up, right?
Yeah.
Could do maybe 160, 10 meters or 20 meters.
Sounds good.
Copy that. 165, 10 meters.
Yeah, let's start with 10, just so I can get a little comfortable with moving with this viz.
So
I will call it in now. Bridge, this is ROV nav.
Can we maybe tilt up and just zoom into the creel
glad you
there,
call it
just
a move if
sort of
you're
a tighter shot of them?
10 meters at 165 degrees, speed 0.2.
Okay, you can try and get what you can get.
Or 10 meters bearing 165, speed decimal 2.
Good copy. Thank
Go
you
ahead,
very
watch
much.
one.
Wow, that is
Absolutely.
that's pretty interesting.
Okay, Nav, did you see that?
fired Niskin 1.
We fired Niskin 1.
Drop
Watch
it up.
lead.
You want to drop the target for us?
So we
Okay,
are
come
going
back
to take
out
a
for
few
the
water
radio.
samples while we're here.
Thank
I
you.
suspect we'll get some biota in those water samples, but we're primarily looking at the water chemistry.
So right now
To
the ship is stationary
Compare
or
this
underway?
maybe to later in
It's
the
currently
dive.
underway. 10
We
meter
have
move
5
at
bottles.
decimal
They're
2
called
knots.
Niskin
Copy.
bottles,
Which direction?
and they're basically
165.
tubes with 2 open
165.
ends.
Okay.
And when
Okay,
we
I'm
are in
gonna-
position, we can
165.
Thank you.
release
Pilot.
the covers to
There.
those ends, and they snap shut
I'm
and
going to try and push through
capture
this
a
cloud
sample
here.
of water.
Yeah,
And
so
they
last
are
time
almost
we had
2
a lot of
liters
krill. The
each.
game was they
And
can't
when
keep
the
up.
ROV comes on
Okay.
deck, we
So you
empty
get
that
serious
water
close,
and very quickly freeze it
race ahead.
so that
They all get
later
left
it can be
dust.
filtered, and
Okay.
You can get a cool
part
shot
of the
for
work
about
will be
10 seconds, and they
to
all catch up
examine
in front of
the
your lights, and they get
environmental
warm.
DNA, the
So it's a
eDNA.
lot of like going and stopping, you know, like getting out there trying to see something.
And we have done this
before they
at several of
swarm
our
us.
dives.
Okay.
Yeah.
I think when we had that last time, we didn't have quite as fluffy a dust.
Yes.
Yeah,
They
I
were
think
also
this one
clouding
will
our lights.
have more
so
in it than
the
any
one
of the previous water bottles.
But if we get out
This-
ahead,
This could be interesting.
Yeah,
maybe the fish won't be
particulate
dusty
and
as much.
biota for
You
sure.
have to watch when you put on your brakes.
yeah, well, we won't be applying many brakes. Hang out, look at these mats for a little bit, maybe, and
Okay.
let me.
Lots of these pockets
me
with the
catch up.
bacterial mats.
If you're
And you can
comfortable, I
see
wanna
the
start adjusting your
reduced
swing arms and
environment
until you're ready.
that lies below
Go
the
ahead.
surface
Go ahead.
sediments,
Okay.
that darker material. Surface sediments are oxidized from the oxygen-rich waters, but below the surface, where the methane is reacting and
Look
there's
at it while flying.
organic carbon, it's a reducing environment.
Can't even see the bottom now with all the
I
life in the
know.
water column.
Yeah, it seems, watch leads, as we catch up to the, like, that the animals kind of stir up the bottom right when they are in the process of kind of clustering around the vehicle.
Looks like your altitude is also increasing.
Yeah, and the
It is,
lights will
yeah.
attract some of these organisms, and then the predators of those organisms
full
also
wide
follow.
video?
And
Yes, you are.
We're
Okay,
like a giant
co-pilot.
lanternfish attracting organisms to the light, although we're not going to consume them.
I'm going to push forward a little bit. I'm streamed out. Oh, I saw a
ROV
bubble.
nav bridge, move complete.
In the white?
Say again, video?
Where did you see the bubble?
It just came up right in front of the camera.
Well, maybe I want to tilt straight down and see.
Oh, it's coming out of the mat. I just
Okay.
saw another. I think. Okay, hold on, stand by.
so as we're getting settled here and getting the pilot situated, I'll talk a little bit about the. Wow, that's a beautiful squid. I'll talk a little bit about the water that we just sampled. So we have these Niskin bottles that Leslie was just talking about, and basically you can use that. It takes a water sample. This is a 1.7 liter sample of water that we know the depth of. It's a tube that basically closes on each end, and it will
we're going to try a
trap
few
that
things
water
with the lights here, Fernando.
at this depth. And
So let's...
so there are many things that you can, many points of data that you can collect from the water samples, such as the
Wasn't quite
chemistry
as
of
bad.
the
turn
water.
off the lower swing
and-
arms.
But one thing that we're going to do with
It
this
wasn't
water
quite
that's
as bad until
probably
the
a little
sediment got kicked in.
less
Let's go
common
full wide
is
if we're not
look
full
at
wide.
the
You are?
total amount of DNA, not just amount, but we're going to quantify the DNA that's in the water column. And so we've been watching, if you've been watching for the last couple of days, we have seen the water column is very thick with organisms and also with marine snow, which is
Kill the
basically
uppers also, the upper swing arms. See what that does.
a combination of
Looks a little better.
dead organisms from the surface or dying organisms. That it can be fecal matter, it can be mucus from organisms that are alive and close by. So it's a kind of a soup of DNA that's
Pushing past
made
the
up
skids.
of all
Let's
sorts
keep
of
those for right now.
input from
Okay,
different
let's do a
parts
quick
of the
zoom
water
on this
column.
bacterial mat.
And so we're going to do some genomics on
Going
that
tight.
to determine the biodiversity in the water column at different depths along this transect. And so we'll probably get signals from the organisms that you can see. We'll get little snips of DNA from those that are obvious, but then there's also
What was that, Fernando?
organisms in the water column that are too tiny to see, and so we can get a good idea of the biodiversity that we
let's
can't see,
come
as
wide.
well as the biodiversity that is obvious
Let's
that we
just turn
can't
them on
see
again real quick
with
and see
our
what it
eyes
looks like,
and
the lower
through
ones.
the ROV's camera. So this is
Yeah, we're going to want those off.
a
You
new
can turn them off and
idea.
film.
Hasn't been done on Okeanos Explorer expeditions
Also,
before,
pilots,
so
looks
this is
like,
a tech demonstration
out there
to see whether
nicely.
this
Would you guys
is
be
useful,
interested
to
in
see
a
whether
Yeah, so let's,
other scientists might
let's try
want
to
to
keep like a
continue to use
0.1
this kind of data into the
continuous
future.
move going here.
I can do that.
And there's going to be
So
a
let's
web
see. You're
essay
looking
about this that should be on the
about
Okeanos or our Windows to the Deep website coming up in the next couple days.
Do maybe like
Well, you could bring us towards our waypoint.
which
Doesn't
is
look
about
that steep.
135. I can put you straight there if that's
Yeah,
what you
that's perfect.
And we also
Great, I'll
took
do,
some
100
water
meter move at
samples during
135
the
decimal 1
midwater
knots.
Can you copy that,
transects
Fernando?
yesterday for the
Yep.
same reason.
Sounds good.
Great.
It's great that we can have visualization of what the water column looked at as well to in tandem with an actual
I think
sample
Bobby
of the
was
water
more
column.
concerned with the uppers. Do you want me to bring them in? Do you want me to test them out to see how it looks?
Yeah, you can bring them back on, see what it looks like.
Okay.
Nav bridge, stand by.
Yeah, let's turn those off.
One of the things
Stone.
we're going to be
Copy.
teasing
Go
out
ahead.
of the genomic data is whether
Yes, I'm ready.
the deep scattering layer has a particular signal of organisms that are indicative
Okay,
of that deep
100
scattering layer,
meters,
and we
bearing
might be able to
135, speed decimal 1.
detect it with just sending a CTD cast down, which is an array of Niskin bottles, and triggering them at different depths, and then looking for the presence or absence of certain key organisms that might tell us for sure whether we're in that deep scattering layer. And when we come and look at it with lights on, like we're doing now, we might disturb some of the organisms. They might,
come in quick. Let's do a
some of
snap.
them might be attracted to the lights. Some of them might avoid the lights,
I'm looking
so
at
we
the
might
sediment.
not be getting as good a representation
best I can
of
give
what
you.
that is
All right,
with
come on
a
up.
big lighted
Too much,
vehicle. But the
too much in the way.
water column sampling without lights that we would do with a CTD cast might be a better
All right, do you copy that
indicator
move,
of
Fernando?
what's really there.
That was 170.
135.
100 meters at 135, so you might be at a slight angle, but should still be generally upslope.
Okay.
Okay, I'm going to center up on you.
Copy.
Just going to try to move a little bit here, watch lead, and get out of this dust.
Right there. That's 135.
And it's worse over here.
Just as an FYI, I've got my blacks completely crushed and
Let's zoom right here, really.
the blacks to maximum. You seem to be missing a piece.
Yep.
An important
Okay, and there's an
piece.
example of some predation. Somebody must have eaten the head off of this midwater fish.
Let's get the self chain here.
And we've seen a bunch of squid around, and they are definitely fans of that type of myctophid midwater fish. A few dives ago, we saw the squid were quite actively
Come out.
eating the tail parts, the rest of the fish, and leaving the head. So this is
All the swing arms are stowed, correct?
That is correct.
a type of predation that we have been seeing quite frequently out here.
Yeah, I'm going to push ahead.
Copy.
And when we first sat down, we noticed that there were
Let's get this guy
long
coming in.
tubes on the seafloor, and those are from quill worms. And this is a siphonophore that we're looking at here. A swimming bell, and then a whole bunch of
Let him go.
other zooids with the pink little balls that travel behind it. And they're a colonial organism.
Yes, and if you look at camera 2, you can see the view from above, from Sirios, looking down on the ROV, and you can see all the snipe eels in the water. They're hovering above us. It's an active community. And all the particulate you're seeing stirred up from the bottom is not coming from our thrusters. It's from all these shrimp and other organisms plowing into the seabed. Ooh, nice view of the jaws.
And that's the snipe eel.
Found some clean water.
Yep, it won't be clean for long. Look at all these stalks here. Let's
And
take
here's
a
some
quick
of
look
those
at the
quill
stalk.
worm tubes that we were talking about, and that's a polychaete. And
Yeah, let's get the
being able to see a whole bunch of these quillworm tubes is an indication that there is a lot of organic matter on the sediment surface, within the sediments. Oh, and what's that? Oh, is that a hagfish?
It looks like the tail. Oh, I see. It's the jaws of the hagfish.
I think it might be.
Wow, we've seen a lot of these from the other end, but nothing this
Do you
close.
have any more? Is that full?
Max.
Okay.
I guess it was several days ago on one of the
Clear.
dives where
Nick, coming out.
we
Let's see if
saw
there's
the
anything else here
hagfish,
while we have
several
it
of
clear.
them, burying into the soft sediment, and they
Some black
basically
sediment
head
up
first
ahead.
dig in, and then there was just a tip of their tail that was sticking out.
Mm-hmm. I guess it was yesterday we did see the very tip of it, but at that point we didn't know it was a hagfish.
Right.
Mm-hmm. Of the jaw end.
There's another headless fish. Yep. Is it headless?
Guess not.
It looked headless to me.
It did to me, Dwayne. It's that little fish lower left.
Let's get the hagfish here.
And there's a hagfish.
Yeah.
I think he has other
Let's do a
things on
snap
its
right here.
mind
Can you
than
come in tight?
worrying about us today.
Is he buried?
Huh. Wonder what this is.
Something living in that tube.
Oh, I guess it's. Is
No,
it a
there's like a shrimp that
fish,
something's
or
eating.
I mean, maybe it's.
I
Oh,
don't
maybe
know.
it is one of the.
They look like the worms
The
coming out of those
worms.
tubes.
Yeah,
And that they may have captured.
one of the shrimps.
Oh my
A
gosh, I think
suggested
it is.
shrimp.
Yeah. They're chewing on it.
Wow.
Takes two. Hold on to it.
Yeah, so this is likely a
Get
quill
closer.
worm, and Amanda says that they love to scavenge, and that they've been imaged scooting along the sediment surface, carrying their tube with them. Yeah, so these two quill worms are going to town
out a
on
little
a
bit.
suggested shrimp.
Yeah, hold that.
Uh-oh,
And then
here
we
comes
have
the
hagfish
hagfish.
in the left side of your screen.
Might
Navigator.
take advantage of their catch.
Go pilot.
Do we have
Yeah,
any
squid
idea
just
where
attacked
the
that
surface
stick.
current's coming or going?
Surface current
We've
currently
seen
is 1-2 out
Several
of
of these squid
120
do this.
Okay.
at decimal
come
7
back in.
knots.
I wonder if they are
So
trying to eat the quill
in the event of
worms.
an emergency, if we were to lose power, we should
They
drift
definitely
away.
picked it up, tossed it
Slow.
over.
Which direction is the bail at?
Coming out of 120.
So
Yeah.
going
Coming out of ones.
going to the northwest.
Yes.
Okay.
And pilots, that's about 25 meters in the 100 meter move.
Copy.
Let's look at this hagfish
There he
real
is.
quick.
That one's
Looking
burying
at you at
in.
There he
60
goes.
degrees.
Doesn't seem to be going very far.
This is the position that we have more frequently seen the hagfish.
Hey, coming up. Is this something different here?
Enemy.
There's a jelly and a cell.
And oh wow, is that a salp or a siphonophore being eaten by a?
That is. I was trying to figure out. Yeah, it looks
don't know
like
if
the
that's-
chain. It does look like that.
It's a cereanthid tube anemone that's eating it, I think. It's hard to say because the way to determine if it's a cereanthid is all these tentacles that are right in the center, and that's where the siphonophore is being eaten.
that Max?
So
That is.
Well, we get a nice close. Oh, he just bit off part of it. I don't know if the tube worms or the anemones can actually bite, but part of it escaped.
I guess they kind of can
Yeah.
bite, but not with teeth.
Not with teeth. Yeah, they don't have jaws.
That on the left.
And there
Oh,
goes the
it's-
siphonophore.
Yeah, about.
So, fortunately, a colonial
Okay, it's getting
organism.
dusty. I'm going to move along. I'm falling behind.
We're not seeing as many of the bacterial mats here. So we just happened to have landed in an area that was quite dense with different marks indicating that past seeps had occurred.
Orange.
Yep.
Hard
go
to
ahead.
tell
Sea star.
how abundant those
Yes.
seeps were.
Oh, and here's one of the mud stars.
Mhm.
Oh, and you can see that quill worm on the left and the right of the mud star. They
Good
have
lasers.
their heads poking out.
Like, there you go.
These are
All right, come in
possibly
tight.
Plutanaster. It looks like the one we've seen several times.
Hey, coming up.
They have pointed tube feet that allow them to dig into this substrate, bury themselves.
Oh, and another hagfish.
90 degrees.
Pushing along.
We had a nice video of one of the mud stars excavating himself by lifting up one arm at a time and knocking off the sediment and raising up on those pointed tube feet. We only had 2 of the 5 arms. It was a long process. We got the
this
gist
fish
of
real
it.
Those are 2 rat tails. Nizumia.
Including the real small one left.
It's coming tight.
Oh yeah, there's a tiny one on the left.
Let's get the one up higher. That is my own dust.
A tiny eel pout.
Red tight.
So I think this is the first time we've actually seen
coming
quill
up.
worms.
Hey, gotta push ahead.
On
Yes, they're definitely new to me. I have not seen them.
And we're now at a depth of 505 meters. Another few
Orange
siphonophores.
stripe at the bottom.
Go ahead.
Huh. I wonder if this is another one of our...
I thought that was the head of it. That's an eel pout on the left.
Yeah, eel pout on the
It almost looks
left.
like it's part of it, but it's not.
I wonder if this is another one of the ribbon worms that we saw yesterday.
There's a lot of organic matter in this area settling down from above, but also because of the lower current velocities here, it has a chance to settle into the sediments.
Really hot.
And provide food for a lot of these benthic fauna.
And so in these canyon areas, we have these steep walls, and one of the walls, the wall that is south-facing, might be influenced by stronger currents from the Gulf Stream, and those that are north-facing are kind of in the lee of the current. And we've been diving more on the lee side, and so one of the things that we'll want to do in the future over time... Oh, look at the quillworm is climbing on this crab. And the crab is eating a shrimp.
Isn't that funny? Hey, that was a good shot of the body of the quillworm.
Yeah.
It was extended. So it looks as though the body of the quillworm has its feet sticking out the top of the tube, so you can see how it mobilizes across the sediment.
We've seen so many different crabs eating usually other crustaceans, other crabs, shrimp.
But a great brittle star feast we saw. That video should be up on the web at this point.
Video's clear.
Yeah. You good, Science?
We haven't seen any brittle stars yet, I don't believe, today.
No.
Have we? Yesterday we saw a lot of their arms sticking out of sediments, very, very similar to this.
Let's get this.
2 days before that, we didn't see very many.
That's another midwater fish, a hatchet fish. Wow, that's great.
Yeah.
ROV nav bridge.
We should have called this a midwater.
I know.
But I wanted to give you an update on the current.
Deep midwater.
Current is still out of a 120, and speed is 0.8.
And then on another dive, again similar substrate, we saw brittle stars everywhere on the surface. So interesting habitat differences in terms of the biota, but not that dissimilar in the substrate.
Right.
All this very fine-grained mud. Here, of course, we've seen that just below the surface is more of the reducing environment. So that might be some of what is causing differences. But just looking at the distributions of the brittle stars, both geographic distribution, but also where they are occurring, either on the surface or buried, or not at all. And then looking at the pout eels, too. Eel pouts. I always want to call them pout eels. They look pouty.
They do look pouty.
You know, those two things have been kind of interesting to see their variations.
Yeah, so once we notice these kinds of trends in differences in animals from canyon to canyon, we can start really honing in on what the environmental variables are that differ between the canyons and how those might dictate what is able to survive and what thrives and what doesn't in certain places.
Let's do a snap here.
And that looks like another myctophid midwater fish.
It's not doing so well. So you see what I'm doing here, Fernando? Just moving back and forth and up the slope. Because if we stay in one spot too long, all the fish start kicking up dust, and the squid, and then the krill start swarming your lights. So
these quillworms are actually
like
quite
this,
beautiful if you can see
moving,
them out of their tubes. They're polychaetes, so they're segmented worms, and they have quite an ornate head.
And then when there's so much stuff in the water, adding more light just makes it harder to see.
Yeah. With nice long tentacles, and we saw those poking out. And their genus is Hyalinoceacea. And possibly tubicola is the species.
Thank you, Amanda Demopoulos, for sending us that link.
Yes.
Nice to see the organism
White
that
structure ahead
makes these tubes that we're seeing so prolific across this substrate.
And again, something that we haven't seen elsewhere.
And it looks like there's a bacterial mat up ahead on the right. Maybe we can take a look at that.
And another hagfish. Ooh. Definitely a very nice bacterial mat here.
Come on in. Just go tight.
And an eel pout sitting in it. And now we look for bubbles and see whether this is an active seepage area.
Right, come on out, partial.
Nope, the squid's sitting in the middle of it too.
It's a big
and
squid.
I guess that these are the Elex squid, the short fin squid that we've been seeing. And if we get the dust to clear, we might be able to get a close-up of the microbial white mat here.
Right. Let's try a zoom here. You want to try turning off my starboard mains, Rhonda, see if that helps. It's probably not, but it's worth a shot.
It's very hard to image these with all the material in the water.
Pause ship move.
No, it's not. It's not going to help.
Copy.
Yeah, you can pause the ship here.
Copy.
We need to tell the squid to stop digging.
I think you're almost done anyway.
Do you want me to turn them back on?
Yeah, you can turn them back on.
Is that a black belly rosefish in the hole behind it? Oh, that's another one of the mystery fish. So we've been seeing this. This is the third dive we've seen this fish, and as far as I know, it's still being debated by many of the fish experts.
We did about 75
which fish it is.
We've not seen any more than its head, so we can't really ID it very easily without seeing the rest of the body.
ROV nav bridge currently holding station.
But it has been consistently the same type of head. So
yep.
These larger burrows seem to be made by that one species of fish. Maybe others, we just haven't seen any indication of others.
Alright, come in on the fish.
And the midshipman was one of the species that it could be, but they are usually much more shallow than this, so that makes that questionable. And then another one was the rye fish, W-R-Y. something like
Yeah, we
that.
gotta move.
It's actually being
There's a
debated on
cloud of fish, I can't
Facebook.
even see.
Yeah, so
Look at the
you might want
swarm
to
in
turn
Sirius.
off one of your main.
Wow.
There's so much biological
Like-
activity that Sirius
Yeah.
can barely see. D2, the ROV that has the camera that you're viewing.
All right, we got to move the ship.
Copy.
Let's
So
do
if
like a
anybody
0.2
wants to
for like 30 meters.
You got it.
follow what's happening in the deep sea with ROVs, the
keep saying
underwater
that.
screenshots. What is their full name? Underwater screenshots, web sharing, something like that.
Go ahead now.
They do a
Get this
great
fish real
job
quick.
of screen grabbing
I am ready
as we
now.
go through our dives, and actually we can get a lot of identifications from the folks that follow this
Okay,
group.
30 meters, bearing 135, speed decimal 2.
Yet another great thing that comes through this telepresence
Right.
availability to the world, and then combined with the social media scientists and
What's interesting about it, Roll?
ROV nav bridge movement
folks who are
if you-
interested in the material are communicating all the time, and then communicating back to us what they think they find.
Let's get the anemone
It's
real
been
quick.
great. We've had several IDs from experts that haven't been on our chat room.
And from all over the world, so there's a Cereanthid tube anemone.
Oh, he just caught something.
Okay.
They have a lot of sticky tentacles along with tentacles with nematocysts. Is that correct?
The cells, stinging cells, and sticky cells.
Do the sticky cells have a name? As cool as nematocyst? I've always liked
I
that
was
term.
I don't know
moving
if
too quickly for that.
I don't remember.
Okay. Great.
Roger
Thank
that.
you.
Thanks, Nav.
Yep.
We've gone about 100 meters so
Do a
far
quick
since we
zoom
reached
here.
the seafloor.
Something just poked his head out. Yeah, let's get the crabs real quick.
Oh my goodness. we
Wow.
have-
Crab fight.
There may be a family.
And those are the Rachinia spider crabs.
There's a snail on the upper right as well.
There's a snail just at about 11 o'clock.
You can go ahead quick.
Another quillworm with its head out.
I don't know if we've gotten an actual ID on this snail. We've seen it a lot. Saw many of them yesterday. So maybe some of
Video's
our
clear.
Right, coming up.
mollusk friends can send that in. Great shot of the quillworm poking out of its tube as well.
And that was the rye mouth, as the other name for that
Oh, rye
fish
mouth.
in
Okay.
the burrows that we've been seeing. And that's the underwater webcams screenshot sharing group. So thanks everybody for all of the work you do to grab screenshots, and then all the other contributors to that Facebook group.
Something big at the bottom.
Yeah, I see it. I think I'm moving too fast.
Shell base.
Go ahead
Andrew
and-
Quattrini from Harvey Mudd College and
Snail.
Bruce Mundy are some of the fish
Quite
experts
large, you
that
should definitely
really
laser
help
it.
out a
You
lot.
know, this might
Lasers
be
are
what
on,
we
right?
saw the other day. If we could just stick with this guy for a minute. Because remember we saw
Another
the
zoom?
bumps
Yeah.
Good.
and we
Better.
saw the siphon, and that's all we saw, but
Well,
it has
to
that
get dusty.
same... And the bumps really are the whorls, I think.
Yeah,
but-
they're just not covered by mud.
Yeah, so I think that is what we were seeing.
It was suggested that we try to collect one
yeah,
of
I
these.
think
We
we
could
might
Could
take
get
that
us.
opportunity to do that.
It would be a good scoop sample, possibly, because I think it's too small to pick up otherwise.
Of course, we may not be able to see it anymore, but Pilot, what do you think about a collection here? Is that feasible?
Bring the ship to stop.
Yeah, it's doable.
Okay.
Stop the ship. That's assuming we can see it.
Yeah. That's
Go ahead now. Easy stop on.
It'll be interesting to see if it is different from the ones we've been seeing at the surface roaming so much.
Shut
Because
all
the two we
lights off
saw,
for a
I
couple
think it was
minutes.
2 days ago, were buried and
We can
not
try
mobile at the time.
Okay, do you want me to turn off the lights? To see if
Holding station.
This one certainly seems like it was recently
Yeah,
buried.
hold
You can see where its
Turn it
trace
here.
begins on the left.
So much action down here.
And we'll also be able to get a sediment sample. Now, certainly it won't be very quantitative. We'll lose
Hey, we're gonna go in the port.
most of
box
it.
here.
Notice that hole in the front.
We'll lose
is
most of
skewing
it
out.
just due to the
We have hydraulics up.
fine grained nature of it. You'll see clouds as we bring the scoop. But at least we can get an idea of what the coarser material is within it. And in this location, it's likely mineral matter that has come off the land and made its way out to this part of the continental shelf, settling down. Combined with some microfossils, some benthic foraminifera that live in
Can
the
you
sediments.
tilt up on my Zeus here?
As well as some planktonic foraminifera, but they are abundant in surface waters,
Keep going.
but there's so much other material in the sediments that
Right.
we call
Come
it
down a
being
little bit.
diluted by the terrigenous material. There's so
Right,
much more
so-
terrigenous material.
It's gonna kick up a lot of sediment when I grab this, so we should try to be
Yes,
ready
and so
to go.
Amanda Demopoulos
Okay. I'm ready to
also
open
thought
the
it would be a good
door
idea to make this
as soon as you
collection.
tell me.
There have been buccinid snails collected further north from here, and that was a cola species, but she's not sure that this is the same one.
Mm-hmm.
And we've had Carol Marshall call in, who's a member of the conchologists. Let's see, what group is that?
In Florida,
Yeah, Broward
Yeah.
County, Florida.
Right, we can open the drawer.
Also saying that this is a
Tilt up on the
species
Zeus.
that they don't have very much information on at all.
Oh,
so
good.
they were excited
Hold
to
that.
Okay.
have
door
a
open.
sample taken. And
Do you want
again,
me
the
to use
sample
right
will
here?
wind up at the
Yeah, you can do
Smithsonian,
that. And I don't know if I'll
and
need
it could be
Get a good
requested
view on HD
by
too.
other scientists to take a closer look at. Oh, I did. Yeah, I put that one in. Yeah, yeah.
Maybe zoom on HD2 and see if it went in there, actually. Well, let's just close it. It's going to take a while to clear up. We can look later if we want. All right, can you look at my arm with HD2?
and that is biosample. It's specimen 02.
Right, tilt down.
We had one water sample previously, so that's 01. This is 02 bio, for those of you who are annotating.
And since we were looking at these sediments, Jason Chater from the USGS said that these are actually very rich in planktonic foraminifera, and terrigenous material abundance is variable as sources. The sources are currently somewhat limited, as most rivers sending material from to the shelf and upper slope are north of here.
Yeah, Jason, I'm sure there are many planktonic foraminifera. The last 2 samples we collected that we sieved at 63 microns had a fair amount of both planktonic and the fine,
go ahead and roll.
fine terrigenous. In comparison to the deeper samples, certainly we have seen by far over 90% planktonic foraminifera. They are very prevalent in the surface waters here, but this is a much lower percentage of planktonic, so maybe I was speaking more in a relative term to the deeper waters, but yeah, certainly as we move closer to shore, we would see higher percentage of terrigenous material as well.
I wonder if that was a cutlass fish that we were just looking at instead of a barracuda. They're similar looking.
Let's do a
long
bump,
silver fish, but
20
the
meters.
cutlass fish is
we
larger.
Keep moving at 0.1.
You got it. 20-meter bump, 135.2 knots.
Wow. Yeah, if anybody wants to see what the water column looks like, look at Stream 2 and Sirius's view of D2, which is totally blocked by all the amphipods and krill in the water column here.
So 20 meters
and
bearing 135.
Surgested shrimps and occasional squid.
ROV nav bridge, move initiated.
Bacterial mat. Let's do a snap. It's going to get dusted really quick.
Another bacterial mat indicating at some point some sort of methane gas leaking. Earlier on a phone call with Carolyn Ruppel of the USGS, she said this is a very leaky margin. I like that term.
Yep.
Big mat on the right.
I see it, Roland.
Sometimes you think you see bubbles, but it's actually just some sediment being just, oh, there are some bubbles, though, on the left. Just saw some come out of that right next to where the tube worm is. Pretty sure that was bubble. It could have been the reflection off of the particles from the lights of the ROV, but may well have been that. They're certainly glistening in these lights, some of those particles.
Yep, near bubbles.
Good. I wasn't making it up. Great. See our first bubbles for the day. Cheryl just stepped out of the room, so she'll be happy to hear. So hopefully we'll stay on this long enough for her to see the bubbles.
So when you talk about bubbles and seeps, how common is this amount of gas compared to a larger or smaller amount of gas?
Well, I've only experienced a few seeps, so I'm not the best one to ask, and Cheryl's sitting down, and we'll repeat the question in a minute. But I will compare this to seeps that we saw on the west coast off the margin, really off of Portland, Oregon area. And they were very rich with methane bubbles periodically. We returned to a site after a couple of days, and there was no activity. And then while we were roaming around the area doing other samplings and other investigations, the bubbles started to emerge from the substrate. And by the time we left, they were in full force again. So seeing just 3 or 4 bubbles at a time here is probably not what is